A protein normally translocates to the nucleus only after phosphorylation of a critical serine residue in its activation domain. Researchers mutate this serine to various amino acids and examine the localization of the protein in the absence of any upstream signaling. Which of the following substitutions is most likely to result in nuclear localization of the protein? So in this situation, there's some signal that leads to a serine gaining a phosphate group. And phosphorylation occurs to amino acids that have an OH group on them, and they take a phosphate group from ATP, and then afterwards, instead of the H on that OH group, we have a phosphate. This can cause major changes to the protein that this amino acid is in, because previously there was no negative charge, but now there is. So think about all of the intermolecular interactions involved with protein folding. Well, those are also involved with protein-to-protein interactions. So if we change something from a polar molecule to a charged molecule, we'll have some pretty major effects in both of those areas. Something that researchers do a lot to study pathways that involve phosphorylation is to substitute one of those amino acids that can be phosphorylated with an amino acid that already has a negative charge. Glutamate, for example, already has a negative charge on its side chain, and amino acids that already have a negative charge on their side chain are called phosphomimetic amino acids because they mimic an amino acid that has been phosphorylated. So without that signal, we won't have phosphorylation of the amino acid at that position. But if instead of serine we have glutamate, we already have that negative charge on the side chain, so it's going to act like it's a serine that has been phosphorylated. So since the phosphorylation of serine caused it to move to the nucleus, if we substitute it with a phosphomimetic amino acid, then that protein should already move to the nucleus, which means C is our answer.